Excellent. Thank you. Good afternoon, everyone. So yes, happy to be here presenting to you all today. I'm going to give a very high-level, broad overview of the types of research that my lab is conducting and our broad interests. I am an epidemiologist by training who got involved in genomics and bioinformatics. As was mentioned, I'm a member of the Genomics and Bioinformatics Cluster and the newly established Pathogen Genomics Core. So this research spans a lot of different areas that I'm going to review. One of them being genomic epidemiology and applications of infectious disease genomics to public health practice. The next, investigating pathogen emergence and evolution, followed by vaccine and drug development, which includes a lot of the computational resources that are rich in the computer science department. Lastly, clinical diagnostics, and then touching more on future directions. So some of my current research that's NIH and industry sponsored, we're really interested in two specific pathogens. One of them is Staphylococcus aureus. It was previously called the superbug. It causes skin and soft tissue infections, bone infections, and is carried normally in our nasal passages as well as on our skin. So we're looking at transmission of this bacteria within the household. And what we do is basically use the genomics of the pathogen to build family trees or phylogenies that we use to understand transmission within those specific settings. Another area of research is a bacteria called Streptococcus pneumonia. It causes something called pneumococcal disease, which is one of the leading causes of pneumonia in the world, as well as a cause of meningitis and blood infection, as well as infections in kids. We currently have a study where we're looking at how many individuals carry this type of bacteria and using genomics to characterize it, identifying antimicrobial resistance, as well as identifying whether these strains are vaccine serotypes, which means that they are able to be controlled by the currently licensed vaccines, or if they're non-vaccine types that are potential targets for future vaccines. So we're looking at carriage in the Orlando area using our clinical sciences teams. And next, which I think ties really closely to Dr. Razavi's presentation, we're looking into bone infections with Staph aureus and what's called osteomyelitis, and also had been heavily involved in the past with the SARS-CoV-2 pandemic. That'll be showing a little bit more of that research. So in the first areas and impacts of genomic epidemiology, basically we're using these tools to track the emergence and spread of a pathogen. This was done very widely during the SARS-CoV-2 pandemic, and we see here two studies that were done. First, on the left, a number of different phylogenies, which are a representation of the genetic distance among strains of SARS-CoV-2 that were collected from cases. And we used these tools to investigate its emergence and spread in Florida and throughout the world. So how many times this particular virus was introduced, and then how many different places did we export this particular variant to. On the right-hand side, we had a PhD student that looked at surface monitoring to SARS-CoV-2. He wandered around campus all year, swabbing different surfaces, looking for the virus, and we correlated the peaks and troughs of activity on the campus from the Student Health Center with the amount that were being detected in the environment, and use that as a form of surveillance to understand high activity areas of the campus, as well as tying those data to our wastewater surveillance that were being conducted at that time. The next area is using these tools for investigating pathogen emergence and evolution. One of these is to characterize transition from carriage to disease. So we have a lot of these bacteria carried on us normally. They don't always cause disease, but understanding why in some instances they do is a particular interest to our lab. One area that we're doing this is looking at intrahost evolution. So from the time that you come in contact from a bacteria or a virus to the time that you spread it on, the pathogen is going through population expansion, evolution. It's driven by host immune system, antibiotic pressure, antiviral pressure, and this forces antigenic evolution or the development of antibiotic resistance. In the past, we've looked at cystic fibrosis patients, for example, trying to understand long-term carriage of Staph aureus and how disease and antibiotic resistance emerges and evolves over years of carriage. Again, right now we're looking at osteomyelitis and bone infections, seeing people who have chronic bone infections and tracking how the pathogen evolves during that time of several years, and understanding how it stays one step ahead of our interventions. Next, we're really interested in informing vaccine and antimicrobial development. The slide on the right was from the seminal paper on reverse vaccinology, where we're now using a different approach to developing vaccines and antibiotics. Instead of just testing all of these different compounds and identifying which ones work against a particular infection, we now are using machine learning and now more recently AI to understand where the drug and vaccine targets are most likely on the pathogen and developing antimicrobials to then target those receptors. And that's been really advancing our understanding of and our development pipelines. For us, we're really interested in tracking antigenic evolution. This is, again, some work we did in Streptococcus pneumonia. On the left-hand figure, we see the population structure of strep pneumo divided genetically into different serotypes that are either vaccine types or non-vaccine types. So some of them will not be affected by the current vaccine. And we are really interested to see what happens when you introduce a vaccine to a population that only targets a specific component of the entire pathogen population, which is very characteristic about all the vaccines that we use. For example, you can think about the flu vaccine only works against certain types of flu, and what happens to the ones that aren't targeted with the vaccine. So on the left-hand side, we have the population before and after drug introduction. And on the right, we have forecasting using different types of evolutionary models where we were trying to predict which one of these lineages or serotypes went up or down after the introduction of vaccine. And we actually did a pretty good job with being able to at least be able to identify which ones went up and down. So these data are really important when we're thinking about how to develop effective vaccines and smarter vaccines, as well as thinking about the inadvertent implications of introducing a vaccine that may increase an antibiotic resistant lineage, for example. So lots of interest in this area and maybe some future partnerships with the department in that area. Last, talking about the area of clinical diagnostics. Well, there's a lot of interest recently in what's called pathogen agnostic genome sequencing, and it's got real implications for clinical diagnosis of infectious disease, as well as in the future prediction of disease severity with particular types of pathogens. The idea here is that instead of this long process, which takes days to send to a laboratory to be cultured, isolated, and identified, we can, in the near future, take a single sample from an individual. We can identify nucleic acid from that sample, which we then sequence rapidly and assemble those genomes and identify any specific pathogens that are in there for computational methods. Through computational methods. And the goal here is to reduce diagnosis of infectious diseases from either a day or even several hours to minutes or hours in the future, and be able to characterize whether they're a known virulent strain of a pathogen or have antibiotic resistance that can then inform treatment. So this is one area where we're working and looking at ways to develop this metagenomic sequencing for the agnostic pathogen development. Thinking about future directions, one, as was mentioned, I'm really invested in expanding genomics infrastructure at UCF. We have some recent investments from my department that we had from some funding to be able to build our sequencing capabilities here. And we're going to hopefully in the future be offering those resources to the department and to the university abroad. That also is going to include an investment in our high-performance computing cluster to be able to support the technology that we're obtaining. So we recently obtained an Illumina NextSeq 2000. We also have technology from Oxford Nanopore that we're using for our metagenomic work. We also, in the future, are aiming to develop our genomic-based clinical tools, as I was mentioned. And one of those areas, again, is trying to use AI or machine learning algorithms to forecast disease outcomes. So I'm hoping to maybe in the future talk to some of you about how to realize some of that when we have this really wealth of clinical and epidemiological data, host and pathogen genomic data, transcriptomic data, taking all of those and being able to identify individuals that are at high risk for severe disease, informing clinical outcomes and really optimizing treatment routines for them. So these are all areas, and again, very high level, very condensed, but happy to go into more depth about any of those. So with that, thank you for the time. There's a recent excursion from my research team, and yeah, I'm hoping to answer any questions.